Silkworms were once used purely for producing silk, and their pupae were often considered waste. And today, those same pupae have the potential to address major global health challenges, offer new modalities for vaccines and therapeutic proteins. If any researchers listening today are struggling with difficult protein expression, whether it's a VLP or membrane protein or complex antigen, I would be very happy to explore how we can support your R&D.
Welcome back to part 2 with Masafumi Osawa from KAICO. In part 1, we explored how silkworm pupae function as natural bioreactors, expressing complex proteins using a baculovirus system. Now we're moving from platform science to product reality. KAICO isn't just offering contract services, they are developing injectable and oral vaccines for both human and animal health. We'll examine their development pipeline, discuss the unique regulatory considerations when your bioreactor is alive, and explore where silkworm-based manufacturing fits into the future of biologics production. Let's continue our conversation. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech.
Let's shift gear, Masa, and let's focus on your product pipeline. What are the kind of molecules you're developing? How well advanced are you in these various molecules? I'm curious to see what you're working on right now.
Our pipeline is designed around a stepwise regulatory strategy, starting with segments that allow for rapid entry and building toward human applications. In livestock, our most advanced program is the TCV2 oral immunization product for pigs, registered in Vietnam as a functional feed additive. So for this product, in real farm environments, it has shown performance comparable to injectable vaccines while substantially reducing labor and stress costs. This provides strong validation of oral antigen delivery using silkworm-derived proteins. In companion animals, we are developing oral vaccines for cats, including FPV, the parvovirus, FCV, flacevirus, and producing purified CPV antigens for dogs. So our aim is to reduce vaccination stress for animals and offer alternatives to in-clinic injections. In human health, our recombinant injectable norovirus vaccine is preparing for phase 1 clinical trials next summer under Japan's AMED SCARDA program. This marks a major step toward establishing insect-based platforms in human pharmaceuticals. Beyond internal programs, we collaborate with partners to express complex proteins and antibodies, leveraging the unique capabilities of the silkworm system. Together, our pipeline reflects a long-term progression from livestock to companion animals to human injectables, and eventually our ultimate goal, human oral vaccine.
So oral vaccines represent an exciting delivery approach indeed, because it will reduce, as you said, the stress when administered. Are there specific antigen characteristics that work better or worse for these oral vaccine applications, and what are some limits?
So silkworm-derived antigens provide significant advantages for both injectable and oral vaccines, and the reasons differ depending on the modality. For injectable vaccines, the key strength is the ability to express antigens that are difficult or sometimes impossible to produce in other systems. This includes complex structural proteins and VLPs of various virus-like particles, which often do not require mammalian-type glycosylation and therefore assemble particularly well in insect-based platforms. The silkworm pupa offers a dense, multi-cell type environment that naturally supports proper folding or multimerization and high-yield expression, which is why we can obtain enough purified antigen from a single pupa to immunize several hundred pigs when we manufacture injectable swine vaccines. This level of production efficiency is extremely difficult to match with the conventional cell culture systems. For oral vaccines, the advantages are even more distinctive. There have been previous challenges with plant-based oral vaccines, such as rice or other edible crops, where expression levels were too low or production costs escalated dramatically when scaled to industrial volume. As a result, despite scientific interest, few plant-derived vaccines have reached commercially feasible. Silkworm pupa, however, function as naturally concentrated bioreactors, and they deliver expression levels far higher than those typically achieved in plants. At the same time, silkworms themselves can be mass-produced at low cost, making them well suited for oral vaccine applications where dosage volumes are much larger than injectables. In our PCV2 oral program, for example, we generally formulate the dose at roughly 1.5 pp per pig, accounting for variation in feed intake and ensuring sufficient mucosal exposure. This is in contrast to injectables, where the purity and potency allow one pupa to cover hundreds of animals. On top of this, silkworm-based production avoids the need for large bioreactors, extensive cultural media, sterile water systems, as I mentioned, and extensive cleaning operations. These factors dramatically reduce manufacturing cost and environmental burden, giving our platform a strong economic advantage, especially in vaccine areas where global accessibility and scalability matter most.
During the COVID pandemic, we have heard over and over how important it is to be able to develop vaccines very quickly. I'm now curious, with the silkworm platform, how fast can you develop a new vaccine? Is this comparable to, for instance, mRNA, or how does it differ?
So as long as the DNA sequence is there, we can produce any kind of recombinant proteins. That's the first thing. One example is that during the COVID, we have also produced SARS-CoV-2 recombinant spike protein. It took just 3 months after the outbreak of the COVID-19. So I think that's one thing remarkable about our platform.
That's remarkable. Very fast. And the advantage I see with your platform as you're scaling out, it's very easy to do a huge scale out and produce massive amounts of vaccines in a short time. Now, on our podcast, we usually focus on human medicine. So I love to have a quick deep dive into the animal side of things, because that's very important. We often forget that there is a huge market for animal health as well. What are the main regulatory differences between the two? Can you just give us the 2-minute version? Obviously, I'm sure there's a lot of more details, but what is the 2-minute version of the differences between human and animal health?
Okay, so regulatory pathways for silkworm-derived products vary greatly between human and animal health domains. So human vaccines follow globally harmonized standards, but because silkworm-derived antigens are unprecedented, we must work closely with Japanese PMDA, the local agency, to define raw material GMP, CMC expectations, and quality control frameworks. In the animal health sector, pathways differ by country, actually. So in Vietnam, our PCV2 product was registered as a functional feed additive rather than a pharmaceutical, enabling rapid marketing entry. Companion animal vaccine follow pharmaceutical frameworks, but with shorter timelines than human vaccines. These differences allow us to pursue a strategic stage progression story, starting with fast approval areas, gathering real-world validation, and gradually building toward more regulated markets.
Okay, I see. It's country dependent. And also what I recall is that there are huge differences between human health and animal health.
Yes. However, for the companion animals, for example, there is a VICH, veterinary ICH. So they are normally harmonized between the countries. However, the product like our PCV2 immune enhancing feed active, since there is no such similar product in the world yet. So the decision depends on the local agencies, how the category should be fitting.
Let's look ahead because you're still quite a young company. What will the future hold? What are your plans? If we look at the therapeutic areas or the product types or the services you offer, what are your next steps?
While vaccines represent our core focus, the silkworm system has broader therapeutic potential. Many antibodies and complex recombinant proteins are difficult to express in standard systems due to folding challenges or instability. Silkworm pupae, with their diverse cell types and chaperone-rich environment, offer an alternative solution for these difficult targets. Sustainability is another strong feature of silkworm biomanufacturing because the system requires minimal water or no large bioreactors and low energy inputs. The overall environment footprint is significantly lower than conventional platforms. This opens possibilities for distributed manufacturing models where production can occur closer to the end user, including in emerging regions with limited infrastructure. In the long term, the flexibility of small batch production means that silkworm may contribute to personalized biologics or rare disease therapeutics.
When do you think will we see the first biologic approved that was produced in silkworm? Do you have any idea? That be 5 years, 10 years?
That's very difficult to answer the question because we are about to enter the phase 1 clinical study next summer, to be more precise. So I don't know how many more years it will take, but it's getting more real, to be honest, because no one believed that silkworm's live body can produce or can be a source of API or vaccine antigens before. But now it's becoming a real thing. And now it's going to be enter phase 1 clinical study means that the product that derived from the actual silkworm live body are now about to be entered into the human body. So I cannot tell.
And that's a major milestone, which shows that you have done the homework and that you have already initial regulatory acceptance. Obviously, there's still a lot down the road, but that fact that you can start phase 1 clinical trials shows that regulatory bodies see the potential and they trust the technology.
Yes. Now the regulatory body accepts the quality control, how we control the quality. And now, which means that it opens the door for us for more pharmaceutical development possibility.
Yeah, absolutely. I agree. You have demonstrated the proof of concept. And once you have laid this foundation, you can build up on this foundation. Yeah, that's wonderful. Before we wrap up, Masa, is there any burning question I haven't asked that you're eager to share with our biotech community?
I think I forgot to mention how much the volume that a single pupa actually can produce, right? So one more good thing about silkworms is that its productivity. So one silkworm pupa size is 2 to 3 centimeters. It can express 10 to 20 milligrams of norovirus virus-like particles. So it's a really good amount. When we purify the protein, it goes down to 1 to 2 milligram per pupa, but still it's a good number. So that the reason why I keep saying that scaling out is one of the very good advantage of our product platform. So that if you want to make 100 milligram orders, then we just need 100 of silkworm pupa, which means that as a size of container, it's just the same size of one laptop. So it's really a tiny amount actually compared to the giant manufacturing equipment. So this is one very good advantage of our platform.
As we're wrapping up, Masaf, what is the most important takeaway from our conversation?
Silkworms were once used purely for producing silk, and their pupae were often considered waste. And today, those same pupae have the potential to address major global health challenges, offer new modalities for vaccines and therapeutic proteins. If any researchers listening today are struggling with difficult protein expression, whether it's a VLP or membrane protein or complex antigen, I would be very happy to explore how we can support your R&D.
Yeah, this has been great, Masa. Thank you so much for sharing your research and also making a way to democratize life-saving therapies, pushing the boundaries beyond what many people believe it's possible. Where can people get a hold of you, Masa?
Please feel free to connect me with on LinkedIn at Masa-Osawa, Osawa.
Fantastic. There you have it, Smart Biotech Scientist. Please reach out to Masa, learn more about their technology. And once again, Masa, thank you very much for being on the show today.
Thank you very much for having me, David. It's my pleasure to be here. Thank you.
Thank you for joining us for this deep dive into silkworm-based biomanufacturing with Masafumi Osawa. From ancient silk production to modern vaccine development, KAICO is proving that nature still has lessons to teach us about efficient bioprocessing. If this episode expanded your thinking about alternative expression platforms, please take a moment to leave a review on Apple Podcasts or your favorite platform. Your feedback helps us bring you more cutting-edge biotech insights. Thank you so much for tuning in today, and I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.